The topic of neoplasia, part 2 lab. The first slide we have is colon polyp. This is the colonic mucosa, and the polyp is an elevation or a projection of the mucosa toward the lumen. This is called colonic polyp. We can divide the polyps into different types: hyperplastic polyp, adenomatous polyp, hamartomatous, inflammatory polyps, and other types. In this example, we have what's called adenomatous polyp, which is this plastic polyp. So these lesions are pre-malignant, and we can divide the adenomatous polyp into tubular adenoma, villotubular adenoma, and villous adenoma according to the architecture of the colonic glands or cells. So in this example, all the colonic mucosa or crypt are arranged in a tubular architecture. All these glands are arranged on tubules, so this is a tubular adenoma. However, if we have villous architecture, so we will call it villous adenoma. And if we have mixture of tubules and villi, then we will call it tubular villous adenoma. So in this example, we have adenomatous polyp, a tubular type. And in the adenomatous polyp, as we said, there will be dysplastic changes. Dysplastic changes, we can identify some of these changes when we compare the normal colonic mucosa at the bottom of, at the base of the polyp with those cells at the tip of the polyp. These glands are darker in color because there is nuclear hyperchromasia, and the glands are more crowded and irregular. So this is the normal colonic mucosa, and this is the adenomatous polyp dysplastic changes. This is a higher magnification to highlight the difference between the normal colonic mucosa and the dysplastic or adenomatous polyp colonic mucosa. In the left side, we have the normal gland. Each gland have basally located nuclei. These are the nuclei which are located at the basal layer, and we have apical mucin in this area. While on the right side we have dysplastic changes in form of nuclear stratification. So the nuclei is no longer at the basal side, but it's distributed all over the layer. These are a stratification of the nucleus. Of the nuclei, and we have also a depletion of the mucin. We don't identify plenty of mucin like in the normal tissue. We have depletion of mucin and nuclear stratification, nuclear hyperchromasia, and also increase in mitotic activity. These dark areas is the mitotic figures. We will see later on in a higher magnification. So we still have similar crypts to the normal tissue. It's very similar to the normal tissue, so this is a well-differentiated type of tumor. This is the colonoscopy of a patient with colon cancer. Colonoscopy is used to screen for any masses inside the projected toward the lumen of the colon. And in this picture, we can identify a bulky mass. This is the mass, which is a colon tumor. And at the periphery of the mass, we have ongoing bleeding. There is a lot of blood at the edge of the tumor, so the patient may also have positive occult blood in the stool. We do stool analysis, and in the stool analysis we can find microscopic RBCs in the stool. So we have two tools for investigation for colon cancer. We have colonoscopy, and also we have the stool analysis test. If we take a section from this tumor and examine it under the microscope, this is a moderately differentiated adenocarcinoma of the colon. We can see this is the epithelial structures or the colonic gland, which is irregular in shape and infiltrative. So the epithelial element of the colon is infiltrating the stroma. So this is a moderately differentiated adenocarcinoma, or less differentiated adenomatous polyp. So the cells is still resembling the normal structure, so this is still be differentiated. And the less differentiated the tumor is, the more aggressive one and more likely to invade the adjacent structures and metastasize. Here in this picture, we illustrate a type of testing we do in tissue is the immunohistochemistry staining. In this example, we have the cytokeratin. We use antibodies for cytokeratin to detect the cytokeratin which is presented at the surface of the epithelial cells. So the cytokeratin is marker specific for epithelial cells. So it will be positive in carcinoma, while it's negative in sarcoma and negative in lymphoma. So cytokeratin is positive in carcinoma because it's epithelial in origin, while it is negative in carcinoma, in sarcoma, and negative in lymphoma. And these immunostains will help us to differentiate between these three groups and determine the cell origin, because in some tumor we have a very poorly differentiated tumor. We can't tell on morphology whether this tumor is sarcoma, carcinoma, or lymphoma. This is another type of carcinoma, which is called squamous cell carcinoma. Squamous cell carcinoma is occurred, for example, in skin and a squamous lining of the esophagus. So in the left side of the picture, here we have the normal squamous epithelium. This is the normal squamous epithelium composed of stratified squamous cells, and we have these basally located cells, and we have maturation also toward the surface and some keratin. While on the left side, on the right side, we have this well-differentiated squamous cell carcinoma. So in the left side, we have the normal squamous epithelium, while on the right side we have the well-differentiated squamous cell carcinoma. We called it a tumor because we have invasion toward the underlying stroma. So we have a basement membrane here on the left side. This is the squamous epithelium, and this is the basement membrane. The basement membrane is separating the epithelium from the underlying stroma, while on the right side we can't identify the basement membrane. The tumor is already invading through to the underlying stroma. So this is the squamous cell carcinoma, and we have abundant keratinization. Abundant keratinization is associated with well-differentiated type of squamous cell carcinoma. So in well-differentiated type of squamous cell carcinoma, we will have abundant keratin, this pinky amorphous material, and we also will have a preserved intercellular bridges. These are the intercellular bridges we will see later on in higher magnification. There is bridges between each two sigmoid cells, and if these bridges is preserved, this go with well-differentiated type of tumor. This is a less differentiated squamous cell carcinoma. It's moderately differentiated. The cells still resemble in somehow to the normal squamous cell, but we have less amount of keratin. This is the pinkish material, which is keratin. So this is a moderately differentiated squamous cell carcinoma, and we have a minimal pinky cytoplasm and keratin deposit. So as we said, in the less differentiated tumor, the tumor will be more aggressive and will be more likely to invade and metastasize. This is a picture for the well-differentiated squamous cell carcinoma. As we said, in well-differentiated type, we will have preserved intercellular bridges. These are the intercellular bridges, which is still there. So this tumor is well-differentiated type. We also can identify the mitotic figure here. This is the mitotic figure. Increase in mitotic activity indicates there is increase in the proliferation of the cells, and this is an indication for malignancy, especially if we identified an abnormal mitosis. Here's another histological appearance for a poorly differentiated carcinoma. We can tell this is carcinoma because the cells is polygonal in shape rather than spindle in shape. So the cell is polygonal, is more with carcinomas. This is a poorly differentiated carcinoma. The cells are variable in size, so some of the cells are very large and other cells are small. So we have what we call the pleomorphism. Pleomorphism is variation in size and shape of the cells, and in some of the cells we have very large prominent nucleoli, like this one. When the tumor is poorly differentiated or show no differentiation at all, we call it an aplastic tumor. This is another example for some tumor common in pediatric age group. Just to compare between this picture and the previous one, as we said, this tumor is showing pleomorphism. Pleomorphism means variation in size and shape. While in this tumor, all the cells are similar in size and similar in shape. So this is a monomorphic tumor, not a pleomorphic. This is monomorphic tumor. All the cells are uniform in size and shape. So we can divide it, the small round, we call it the small round blue cell tumor because the cells are small or the same size, rounded in shape and blue in color. And in this category, there's a different type of tumor. For example, we have leukemia lymphoma, which arises from blood, marrow, and lymph nodes. Neuroblastoma, which is also a small round blue cell tumor arising in the adrenal gland and extra-adrenal ganglia. Also, medulloblastoma arising in the cerebellum, retinoblastoma from the eye, Wilmsen tumor we see in the kidney, and Ewing sarcoma, it's also a small round blue cell tumor of 1. This is another histological appearance for carcinoma. I can tell this is carcinoma because the cell is more rounded or polygonal in shape rather than a spindle, and we can still identify some pinkish material here. And so this material is keratin. So this tumor is squamous cell carcinoma because we have minimal amount of pinky keratinization. So this is a squamous cell carcinoma, but it's poorly differentiated type. We can also identify the mitotic activity here. Normally, we can see mitosis normally in the skin and in the bone marrow and gonads and GI tract. While if we see increased mitotic activity in other side of the body rather than these 4 areas, we have to think of malignancy, especially if we see an abnormal mitosis. This is the example for abnormal mitosis. This is abnormal mitosis because it is tripolar in shape. It's a tripolar in shape, so this is abnormal mitosis. And abnormal mitosis is always abnormal. We cannot see the abnormal mitosis in the normal tissue. So abnormal mitosis is always indicating malignancy. We have also another mitotic figures here. Now we will talk about sarcoma. Sarcoma is the malignant tumor of mesenchymal origin. As we said, the carcinoma is the malignant tumor of epithelial origin. However, the sarcoma is the malignant tumor of mesenchymal origin. And it differs from carcinoma in that the sarcoma cells tend to be spindle in shape, like the one here in this picture. The cells are more spindle, so this is a sarcoma. And also in this picture, you can easily identify some huge cells. There is some large cells, while other cells are very small. So this tumor is also very pleomorphic. So this is a very pleomorphic sarcoma. This is a higher magnification for the same tumor. We can identify the pleomorphism. We have very large cells and other smaller cells. So there is variation in the size and shape of cells. So this is a pleomorphic sarcoma. And this type of tumor in this example is actually poorly differentiated, and as we said, the poorly differentiated tumor is called anaplastic. Here's a higher magnification to highlight the mitotic figures. Here we have increase in mitosis, and one of the mitosis is actually abnormal in shape because it's tripolar. So this is the abnormal mitotic figure, which always indicate malignancy. This is an immunomarker specific for sarcoma. In a previous slide, we mentioned that the carcinoma is specific immunomarker is cytokeratin. So cytokeratin is specific for carcinoma, which is epithelial in origin, while we have vimentin, which is specific for sarcoma, which is mesenchymal in origin. So vimentin positivity is seen in sarcoma rather than carcinoma or lymphoma. In this example, we have these cells which take the brownish stain, which is vimentin positive sarcoma on the right side, while in the left side we see this vimentin negative because it's white in color. They didn't take the stain, so it's vimentin negative. These areas is actually skeletal muscle bundles. All these are skeletal muscle bundles, and these are normal structures, so they are vimentin negative. While the tumor cells are vimentin positive, and the tumor cells is infiltrating through the skeletal muscles. Here is a gross appearance for a malignant bone tumor. This is the femur, and at the proximal femur, this is the head of femur, and at the proximal side of the femur, we have this mass. This mass is arising from the cortical area of the bone, from the cortex, and extending outside the bone to form a mass. And this is sarcoma of bone. Sarcoma of bone is called osteosarcoma. Osteo means bone, and sarcoma is the malignant type of tumor. So we have osteosarcoma of bone in this picture. If we take a cross section, if we take section from this tumor and examine it under the microscope, we will get this appearance. This is the sarcoma of bone, or osteosarcoma. It's a spindle cell tumor. These are spindle cells, and these spindle cells produce osteoid material. This is the osteoid material. This is the pinky amorphous material. All are osteoid. So this is an osteosarcoma, which is sarcoma producing osteoid material. Another example for sarcoma, which is the liposarcoma, which is sarcoma of lipo mean adipose tissue or fatty tissue, and sarcoma is malignant tumor. So we have liposarcoma, which is the malignant tumor of adipose tissue, different from the lipoma, which is the benign tumor of adipose tissue, is called lipoma, while the malignant type is called liposarcoma. In this example, we have this large mass of fatty tissue. It's measured 15 centimeter in the greatest dimension, and the cut surface of the tumor is very homogeneous, yellowish in color. It's very difficult to differentiate between this tumor and the benign lesions, because the tumor is very well differentiated in this example. This is a well differentiated tumor, so this is indolent tumor. We can just differentiate between the benign and malignant, well differentiated type by histology. We can't differentiate in gross appearance. So the liposarcoma is the malignant tumor of adipose tissue. The most common site of liposarcoma is retroperitoneum and thigh, and they occur in middle-aged and elderly people. If we take section from this tumor and examine it under the microscope, we will get this appearance. This is the liposarcoma histological appearance. We can see that there is two different type of cells in this slide. We have larger cells and we have smaller cells. The large cells is called adipocyte, which is the benign type of cells. Benign adipocyte, which is the benign fatty cells, while in the smaller one is called lipoblast, which is the malignant cells or the neoplastic cells. So the liposarcoma, when it's well differentiated, it will be composed of a mixture of lipocyte, of lipoblast and adipocyte. This is a higher magnification. The larger cells, as we said, it's the benign one. The larger one is the benign one, which is called adipocyte, while the smaller one, which composed of vacuolated cytoplasm and very folded cytoplasm, all these smaller cells with vacuolated cytoplasm is lipoblast, which is the malignant cells, while the larger cells is the benignish one, which is called adipocyte. This is the last slide. Have you any question? Okay, يعطيكم العافية.